Okay. Can you give me a
Ready?
feedback? Yes,
Yeah.
I am. Okay, so can I speak?
Yes.
Okay. Hello, good morning, everyone. And thanks, Scott. Thanks also to the other organizers, Andrea Diaconescu, Milad Bancarani, and John Griffith, for putting on this great event of CCNS for this year. And as I said, good morning or good afternoon or good evening from wherever you're joining in. This is Maurizio DePitta speaking to you from the Basque Center for Applied Mathematics here in Bilbao, Spain. And as a chair of this session of synaptic physiology, I was asked to provide you with a quick background in order to later on also follow up more closely what my colleagues are going to present in this panel. So I should start with probably rhetorical questions. Most of us in this audience I'm talking to are familiar with the concept of synapses. So let's start with a very simple question. Why in the year 2021 should we still study synapses? And to answer this question, let me just make a simple analogy. Let's take the single bee. Once we have one bee, we cannot produce or make honey, right? We need many bees together that swarm in thousands of entities that are able to coordinate foraging and then regurgitation and then storaging of the honey in order to make this precious nectar of golds. And so the bees in their swarm are like this nervous net of a hive individual. And the analogy goes that neurons are like bees. Our single neurons alone cannot make up our brain, but we rather need them to wire together into functional circuits by means of what we now call synapses, a term that derives from Greek and literally means to fasten together. So the idea that functional circuits made up by synapses are at the core of our being, a concept known as the synaptic self, and by incidentally, it's also the title of one of my favorite books on the subject, along with another one by Reynaldo Rinas, Eye of the Vortex. It's really literally the notion that we are what we are wired by. And so at the center of this simple concept is the fact that if we want to understand brain function, we need to unravel what are the neuronal functional circuits of our brain. And this eventually breaks down to understand what we call the synaptic space of our brain. The proof of concept of the synaptic self comes looking at the pathology. So several cognitive deficits associated with different disorders are correlated with alterations of the so-called spine density. Spines are just anatomical proxies in neurons for the synapses in our brain. So a fundamental example is dementia and Alzheimer, right? What happens in the patients in the patients with Alzheimer is that there is a deposition of plaques on synaptic sites, which literally breaks the circuits and tear apart the neurons. And we look inside the circuits and we see that synapses are lost. The idea that dementia is connected with the fact that our circuits are not any longer able to transmit information. On the other hand, if we look instead in autistic children, we observe an excess of synapses, perhaps associated with the fact that autistic children are often observed in this kind of repetitive behavior, or they are able to over-amplify the synaptic, the stimuli that they receive from the world. And yet another example is a schizophrenia. Here again, although whereas the Alzheimer was in the hippocampus, schizophrenia here is in the prefrontal cortex, we still observe a lack of synapses. So there is an important difference, however, between Alzheimer and schizophrenia in this case, right? Which makes up some essential notions on what makes up our synaptic self. Literally, not only synapses number is important to define what we are, but also how this synapse number changes, the direction of this change at different stages in our life. So decreasing synapse numbers, for example, during adolescence result very likely in schizophrenia, whereas a decrease at later stages in our life results in dementia of Alzheimer. So the so-called synaptic space, as I mentioned it before, is something that we understand as to be dense, but also something that changes in time. Now, when we want to understand what is this space mathematically, we can resort to some kind of seminal work back in the 90s on the macaque brain by Van Hessen and colleagues, and later on in the human brain by Ola Sporn and other mathematicians, that look at brain circuits just as graphs. A brain circuit here on the macro scale, but the same happens at the micro scale of simple neurons and synapses, are just the graphs. The nodes of these graphs are obviously the neurons, synapses are the edges. And so let me introduce this very simple mathematical tool, the connection matrix or adjacency matrix of the graph, which will become soon the weight matrix of our circuit. This matrix has as many columns and rows as the neurons in our circuit. And an element is one, whether at the coordinates of a column and row, there is a pair of neurons that is connected by a synapse, or zero otherwise. Of course, when we look at just the graph as entities of connected nodes, this matrix is symmetric, but in reality, synapses are oriented objects. So there is always a presynaptic neuron that connects a postsynaptic neuron. So this matrix becomes asymmetric, right? And also the synaptic weight, the strength of these connections is not just 0 or 1. The strength can be modulated, can be various, and usually along an entire spectrum of, continuous spectrum of values. So you understand that understanding synaptic and brain circuits goes on the characterization of the structure, somehow, these weight matrices. And so a lot of effort in this respect is done in the direction of understanding what are the modes that ensue from this knowledge of the structure of the matrices and how these modes or eigenmodes reflect at the level of networks. So some seminal work back in the 1980s was looking especially at random weight matrices of networks of populations of single cell type neurons. And what they found, this work was by Sompolinsky and Crisanti, what they found is that the average synaptic weight has to exceed some kind of threshold value that without loss of generality we can pose to 1 in order to observe what is defined as chaotic dynamics, which in this kind of models represent essentially the irregular firing observed in cortical networks in our experiments. Now, an update of these of that theory that you see here, instead, consider the more realistic scenario that brain networks are not just produced by one type of cells, but different cell type populations. And so it tells us that in that case, it doesn't have to be considered just the average synaptic weight as demarcated here by the red circle, but rather we have to look at the block structure of the ensuing weight matrix and consider the leading eigenvalue of that matrix in order to understand whether or not there might be either ensemble or subpopulations of cells that are capable of displaying this kind of irregular chaotic activity. And even farther, an example of this analysis can be taken looking at the contribution of recurrent local synaptic connections within same cells within cells within the same population, and instead long-range, non-local feed-forward synaptic connections from one cell type to another. So what is shown here is, if I term S the strength of the recurrent connection and G the one of the feed-forward connection, is that as soon as S is slightly above 1, the original limit in the random one-cell type population, then in general, the network activity and the individual units relaxes to, you see, random fixed point structures, which is something expected in our idea. But there is always still, for lesser, weaker values of strong synaptic recurrent connections, and yet sufficiently high values of feed-forward connections, the possibility for the emergence of irregular firing. If we move forward, then an example of structured connectivity comes from what is known as the Dale's principle, namely the fact that neurons are excitatory, are distinguished into excitatory and inhibitory, okay, depending on the action that they exert on their postsynaptic target, but they cannot be both. This brings up then the question of what is the relative contribution of synaptic excitations versus synaptic inhibition on the overall network activity. And what you see here is a figure from a landmark paper by Nicolas Brunel back in 2000 that suggests or prove by a very famous model that cortical networks might likely operate in a regime where inhibition is strong, such that it balances out the effect of recurrent excitation and feed-forward excitation that otherwise would result in this highly synchronous epileptic form-like activity. And in this balanced regime, the interplay between the inhibitory strength of synaptic connections as well as that of excitation from other regions of the brain is able to account for a very wide range of dynamical possibilities for the network activity, ranging from asynchronous to more synchronous versions of neuronal firing, as well as to highly regular to irregular firing, okay? One caveat of this balanced network model is, however, the fact that if we look at the transfer characteristics of this network as a function of the input stimuli, okay, the characteristics tend to be linear, which is at odds with what we observe in real world. Our brain is usually known to perform nonlinear operations, such as nonlinear summations of two stimuli as we get from the eye and from the ears, for example. So one other topic of investigation is trying to understand how we can retain the balanced conditions that account for the irregularity of cortical firing, and at the same time observe the emergence of these nonlinearities. And so it can be shown that still the balanced network model is still valid and account for these nonlinearities when we come down into realistic combination of either the degree of connections, namely the number of synaptic connections per excitatory neurons, or the strength of both local excitatory and inhibitory synaptic connections, as well as the one of afferent external connections. Another way to look at weight matrices in synapses is in search instead of patterns of synaptic weights as an encoding of some information, in particular, basically a memory of some object. In this case, this matrix, for example, could be regarded as a memory of a circle, right? So there are many questions that you can ask in this context, and two of these questions are, first, how many synapses do we need in order to memorize something? And so one of the approaches that I follow, for example, is to regard synapses not as just binary objects, but to think that they can exist in multiple states, possibly due to the underpinning complex biochemistry that dictates their function. And so it turns out that depending on how you envisage these different states, then, for example, you can come up with situations in which the memory capacity of your network may scale linearly, okay, on the log-log scale, with the number of synapses that you have. So now we have about 1,000 trillions of synapses in our brain, the estimates. So you make more or less an estimation of how much we could learn according to this. Another dual approach is instead to ask whether our brain networks, our circuits, are optimized to store information. Optimization here is in the sense, is our brain capable of storing at maximum capacity? So the problem is just an optimization problem. And in particular, we look at our synaptic space and the volume occupied by a memory item, which is this triangle in our synaptic space. And we look at what constraints our model requires in order to have the smallest volume possible in the entire synaptic space. So Nicolas Brunel, again, a few years ago, came up with quite an elegant demonstration of the fact that there is indeed evidence or support theoretically to the idea that our brain networks may be optimized to work at maximum storage capacity, showing that if this is the case, then the distribution of synaptic weights, represented here by the black line in the theoretical model, indeed matches that observed in the cortical layer 5 synapses. I think it was in the somatosensory cortex. At the same time, the synaptic weights are in general sparse, with a prevalence of many multiple silent synapses, and bidirectional coupling between neuron pairs is preferred against simple one-way connections. Still, one problem of memory is to keep it, right? And so the stability of memory depends on the stability of our synaptic connections. And so if we had to look at spine dynamics in neurons in our brain, you would probably be quite amazed to figure it out that there is a high rate of turnover, that spines actually disappear or may appear in multiple flocks at different stages of our observation, and also they vary in size, meaning that the effective synaptic strength of the excitatory connections that the spine represent is changing. So the question here is how our brain is capable of storing information if the underlying circuit is constantly changing. And what I'm showing here is some ideas that Gianluigi Mongillo, who I believe is going to talk about this later on in this panel, came up showing elegantly that our cortical networks, again, the balanced network model, can robustly account for rewiring of basically the majority of excitatory connections. And brain networks have mostly excitatory connections, and to a lesser extent, instead, of inhibitory connections. In particular, okay, inhibitory connections are the one that dictates whether or not our brain is robust against changes, and this limit is hit in the moment in which we lose the balance between inhibition and excitation. So this pretty much makes up the notion of synapses as elements that connect the neurons. But I haven't told you much still about what is the biophysics of these synapses, what they are, these things in terms of a physical system. And so the synapses that we are dealing with in today's session are chemical synapses. It's true that they transmit electrical signals from a presynaptic neuron to a postsynaptic neuron, but they actually require an intermediate step of chemical translation. The action potentials arrive briefly, release some neurotransmitter resources, or in the vesicular form, that they travel to the postsynaptic terminal where they target some kind of receptors. And these receptors eventually either depolarize, as in the case of glutamate, or hyperpolarize, as in the case of other neurotransmitters. Postsynaptic terminals depending on whether the neuron above, the presynaptic neuron, is either excitatory or inhibitory. Now, there are two different types of synapses in our brain. The one that you see here is the bipartite synapse, okay? But there is also the tripartite synapse, which is a bipartite synapse with the astrocyte elements standing by and dropping the two terminals. As you probably figured out, tripartite synapse and bipartite synapse, right, are fairly different in their biochemistry, with the later being a little bit more complicated. Now, the aim of this figure is to create a wow effect, or maybe some disgust or even frustration, depending on which group you want to be in. The message here is, I'm not going to go into the details of these pathways, but it's worth to remind that 95% of the pathways that you see here has been basically discovered in the past 25 years. And still, despite the surprising biochemical differences, modeling here is advantageous because it allows to essentially describe both tripartite and bipartite synapses in an alike fashion. Namely, we can look at the generic synapse and distinguish essentially between at most 4 compartments: the pre- and postsynaptic compartment, separated by an extracellular space compartment, which accounts both for the synaptic cleft and the perisynaptic space, and a 4th compartment, which is the astrocyte that may or not be there, depending on the type of synapse. And once we look at the relationship of the postsynaptic current as a function of the input stimuli, eventually this input-out relationship can be written as a function of these 4 compartments at the most generic case. In the simplest scenario, however, we can use what is known as the quantal model of synaptic transmission, which tells us essentially that synaptic transmission can be just described as the composition of 2 components: presynaptic release probability of quantal of neurotransmitters, of vesicles, and a postsynaptic current component, that is the postsynaptic end plate current ensuing from one single vesicle. And then we should consider how many vesicles at most our synapse is releasing. This model was put forth originally by Katz in the 50s, who was also one of the first to observe that synaptic transmission is highly variable. The advantage of the model, and I believe Richard now is going to speak more about the sources of the variability. In this case, the advantage of this model is that when Katz decides to fit this model by a binomial distribution, the input-out relationship is just the product of these 3 quantities, n, p, and q. And once we look at the variance of synaptic current, the variance is parabolic function with respect to the mean, which allows us to know essentially, or to estimate, that we don't have most of the time, the number of release sites. A caveat of this model, nevertheless, is that most of the time, n, p, and often q change. This brings up the notion of short-term plasticity, namely the fact that synapses transmit in a way that is dependent on the recent history of their activation. The story goes in this sense that if we look, for example, at the release probability, okay, according to the Katz model, this release probability can be recast as a function of 2 pools, one of relatively releasable neurotransmitter and another one of synthesis of neurotransmitter. Now, when I release, I obviously subtract some neurotransmitter from the readily releasable pool, which requires the reservoir to resynthesize this transmitter to be supplied again to the relatively releasable pool in order to account for continuous or sustained transmission. It's obvious that in the moment in which I release too fast or more than what I generate, my synapse is not able any longer to account for transmission and quickly depletes. At the other hand, when the synapse is able to accumulate, the more I excite for some kind of mechanism that I'm not going to enter into details, I start releasing less, and then as the stimulation goes on, I release more. These two mechanisms, decreasing with frequency and increasing with frequency of stimulation, accounts for traditionally short-term depression and facilitation. And there is something remarkable to say about these two mechanisms. First of all, because synapses with different short-term plasticity accounts for different filtering properties. Climbing fibers in the cerebellum, okay, are depressing synapses, and accordingly they are preferentially transmitting stimuli at low frequencies of incoming presynaptic spikes, differently from parallel fibers that instead tends to transmit frequency at higher rate. This is because parallel fibers are afferents from granule cells, which are known to fire much faster. And Schaffer collaterals instead in the hippocampus, they encompass both mechanisms. An important aspect of these filtering properties is that they are assumed to be fixed. But once you consider instead the tripartite synapse, the filters changes depending on the state of activity of the astrocyte, literally essentially enabling your connection to work like a radio receiver and to tune in on the specific frequency features of your incoming spikes. Now, one issue with these models, and always, is we need to assure not only stability of connections, but also stability of the functionality of these connections. So where is the neurotransmitter coming from? Let me just quickly recap on this and then end towards the last couple of slides. So we usually think that our glutamate, for example, at the cytatory synapses, comes from the blood, right, the circle, through the glucose, and that is then glycolized into pyruvate and enters the mitochondria, where is turned eventually into glutamate. Or it could be just available in the intracellular space due to the uptake of transporters. But there is actually another pathway in tripartite synapses that comes from the glutamine. And in particular, there is a cycle whereby our tripartite synapses, the glutamine that is released, okay, enter the astrocyte, is translated into glutamine through this enzyme, the glutamate synthetase, and then shuttled back to neurons. So there is the possibility that these synapses, when the astrocyte function is impaired, the transmission also is impaired. And it turns out that in the hippocampus, again, the same Schaffer collaterals, when we block this enzyme, okay, and we stimulate the synapses with intermediate bursts of high frequency, which are in the frequency of sensory stimuli, you see the synapse performance in terms of how much we transmit is degraded, but can be rescued nevertheless once we supply glutamine in the extracellular space. Now, I can't even finish talking about synapse physiology, mentioning also what we know synapses for. The fact that we learn by synapses. And the way we learn, well, it can be in many ways. One famous paradigm, probably one of the simplest, is Hebbian plasticity. Now, namely the fact that the coactive neurons are supposed to strengthen their connection. Back in the days, Henry Markram has shown us that this actually requires stringent timing between pre- and postsynaptic firing, namely only when the presynaptic spike precedes within a narrow time window, we observe potentiation, otherwise the synapse is weakened. And then there are two potential topics that are touched in this regard. The first is if we know what are the potential biophysical mechanisms, what are the spike time independent rules? And what you see here is just one of the latest additions to the models in the field that focus on the fact that many curves of spike time independent plasticity can be accounted for by the consideration of the intracellular calcium dynamics, both pre- and postsynaptically, and more recently, extrasynaptically. And the other way around is the dual question: can we say something about what are the biophysical mechanisms that underpin learning once we know the learning outcome? This is what Subin is going to talk later on. And here I just show, for the sake of completeness, one interesting hypothesis put forth by Urbanchik and Sen in Bern, namely the fact that our neurons could be inherently performing what is called supervised learning, meaning there is always a soma and a dendritic compartment. The dendrites and the spine on the dendrites are those that have to be modified or learn the signal, and the soma, by electrical interactions with the dendritic compartment, is somehow able to minimize the error between what it generates and what is expected to generate based on the teaching signal that these synapses are obtaining. Finally, well, everything that works through synapses require energy. And the sources of the energy comes from the mitochondria. But mitochondria themselves require fuel to function. So where does fuel come from? An influential hypothesis, still is pending demonstration, but I believe in this sense, Julia in the keynote address is going to show us more, is the so-called astrocyte to neuron lactate shuttle. The idea is that in conditions of high frequency or release, or when energy demands the synapses are considerable, the released glutamate uptaken by the astrocytes, it's also taken inside the cell together with the co-transport of sodium ions, which cannot be extruded from the astrocyte unless the sodium-potassium pump is at place. Of course, the sodium-potassium pumps is an active mechanism of extrusion which consume ATP, and the cell senses the consumptions of ATP, triggering glycolysis that eventually produce lactate, which is then shuttled back to neurons and made to them as an available energy substrate. So it turns out that in probably what is the most important so far evidence for this mechanism, when we use mice and we train them in an inhibitory avoidance task, namely they are in two chambers, one of them is electrified, and the mouse enters the chamber, learn through an electrical shock that this is a chamber where it doesn't want to be, and we test them after 24 hours and several days after. And we see that the more he stays in the safe chamber, the more he remembers. If we block the transport of lactate from the astrocyte to neurons, well, it turns out that the mice that have this blocked transport apparently, once repositioned in the safe chamber, quickly enters the electrified chamber, suggesting that this lactate shuttling from astrocyte to neuron could be a fundamental mechanism in maintenance of long-term memories. Okay, I conclude the several declinations of this short tutorial talk, starting from the synaptic self, right? I try to recast the problem of why we need synapses, because they make up the functional circuits of our brain. They store the memories, and what we're going to see in this panel are chemical synapses. But the questions are multiple. How can we use synaptic function if it's inherently stochastic? How do we learn, and how do we preserve these memories thanks to the stochasticity of volatility? So the four speakers that will follow, will discuss these aspects more in the details. And before concluding, I would like to share happily that I am proudly joining the Cranville Institute in August. So I am opening my lab on neuroglia interactions. I would definitely prompt you to follow me on Twitter or inbox me if you are interested in collaborating. I have currently 3 positions available. One PhD project whose deadline is expiring at the end of June, And I also willing to recruit postdocs. So in a week, we have the deadline for the Human Frontier Science Project, letter of intent. And by September, whoever could be interested in applying for a Marie Curie fellowship, working on neural interactions, please inbox me. And finally, if you want to know more, I definitely prompt you to follow up with the two trainees talks that are members of my group here in Bilbao. Evan Kressel-Quay, who is speaking tomorrow at 9 in the morning, giving you the coffee salutation. And right after, Giulio Bonifazi in the session 3 of trainees talk in the morning. Said that, I would be glad to quickly take a couple of questions and then leave the stage to the next speaker in the panel.
Thank you so much, Maurizio, for an excellent talk. We'll have to move on to the next session, but we're taking all your questions for the panel discussion. Okay. Thank you again for a great presentation.
So I go to the other session, right?
Yes. See you.
Okay.
See you there.
Thank you.